Good evening, my dear students. Hello, how are you? How are you all? So, I'm back with your HFA topic today. Today, I'm going to discuss about cellular level of organization. Okay. So, this is the main topic in HAP, in human anatomy and physiology, where in human anatomy and physiology, you will be studying about common anatomy of all the organ systems and all. Okay. Anatomy means what? Do you know what do you mean by anatomy? Anatomy means you are going to study the internal structure of a organ. Okay, internal structure. And what do you mean by physiology? Physiology. Physiology means you are studying about the function of that particular organ or cell or tissue, whatever it might be. Okay. Now, so what is the importance of this HAP in your studies in your B Pharm? Do you have that knowledge? And I uh, I hope so. You have started with your syllabus, right? So what? Why are you studying this HAP in pharmacy? Pharmacy means the study of drugs, right? So you are studying HAP here. So my dear students, HAP is a very important subject in all the medical fields. In all the medical field, whether from MBBS to nursing, pharmacy, and whatever what type of medical field you choose, you this subject will be common for everyone. Here you will be studying about the basics of a human body. What are the basics? What are the basic terminology we are using? What are the different things which are present in a cell, which are present in a tissue? What are the functions of those cells and tissues? Once you are perfect with all these things, then only you will be able to excel in your specialization. Like example, if you are studying MBBS, then only you will be able to study about the disease, what is happening in a particular cell, what is happening in a particular tissue, and how to diagnose a disease. All these things will be depending on the anatomy. Okay, if you have done your if you are doing your dental, then also anatomy is important. Okay, and if you are doing nursing, then also first they will study anatomy, then only uh, they will be studying what kind of patient care they have to take, what kind of drugs have to be given. Now coming to pharmacy, now this HAP will be a foundation for your pharmacology, which you will be studying in your future semesters. Okay, it is a foundation for pharmacology. And what do you mean by pharmacology? It is a study of drugs, which is a core subject of your pharmacy. Okay, so to understand pharmacology, first you should have a strong foundation, a strong knowledge of HAP, human anatomy and physiology. Okay, so even when you go outside after, now don't think that this subject will be present only in this semester. एक बार पढ़ लिया फिर छोड़ दिया, फिर we don't need it. No, it is not like that. This HAP will be there for you for your whole career. If you are in pharmacy field or if you are in any medical field, okay, the whole career will be depending on this HAP, because these are the basic things you have to know. You have to know. When you want to be in a medical field, you have to know about these things. Okay, so even if you attempt any exams which are government exams, or if you are attempting any interviews, or any written test will be there for any interview. So in written tests, in exams also, the most of the questions will be from HAP and pharmacology. So never ever neglect this subject, and study this subject with complete willingness. Okay, don't take it as a pressure, don't take it as a stress, otherwise you won't be able to remember. Okay, okay, shall we start with the content then? So how many of you feel, or how many of you know about the cell structure? How many of you know about the plasma membrane? Any idea about these things? Already intermediate you have been studied this, right? You know about a cell, plasma membrane. Then what? Why we are studying again means now you are just brushing up the knowledge which you have gained in your intermediate. Obviously, after intermediate, might be you have got so many holidays, and might be you thought that chalo inter to khatam ho gaya. Now we will leave everything, and might you have washed your brain nicely with all the detergent and soap, and you have cleaned your brain, right? So just to refresh the knowledge, to refresh, you just brush up the knowledge, and we are studying about the cell from the beginning to the deep depth. Okay, so in this today we are seeing what contents we are seeing is, first we will see about the cell structure and different parts of a cell. Might we have drawn this diagram so many times, right? But still we are going to study it. Then you are having a plasma membrane, then cytoplasm, then nucleus, then transport across the plasma membrane, how it occurs, and finally you are going to study about the cell signaling system. Okay, so coming to the cell structure. So this cell structure, what do you mean by cell? A cell is a basic living structural and functional unit of a body, right? So it is a basic thing, right? For how, if you want to, you know, building blocks, right? Each block you will be having. With that block, what you will do? You will be joining the blocks and you will be making up a building, right? In the same way, here also, you are having cells. The cells are the basic things. This is a basic unit of a body. Now, this cell, all the structurally similar cell, or the cells which are having common function, all will be bound to form a tissue. Okay. Now, this tissue, next level of organization will be your muscle. Okay. Then finally, organ. So, like that, there will be different levels of organization. So first, before cell, what will be? Can anyone say? Before cell, before cellular level of organization, what things you will be studying? Before cellular level of organization. Anyone? Can anyone say this? We will be studying about different chemicals. Like how is a cell made up of? What are the different ions are present? All those things. Okay. Then comes your cellular level of organization. This is the basic one. Okay. Structurally, it is a basic one. Functionally, also, it is a basic one. Okay. Structural unit and also a functional unit of a body. Okay. Now, there is a science which, when we study about the cells, the science which deals with the study of cells will be called as cell biology or cytology. Okay, cell biology or cytology. Now, coming to the structure of cells, we have three main parts of cells. Okay, the first one will be called as plasma membrane, which is the outermost layer of a cell. Then you are having cytoplasm, which is inside the cell, and in that cytoplasm you will find nucleus. Okay, so these are the three major parts of a cell. Now, in this again, we will study what are the different parts of in this again. So first thing is you're having is plasma membrane. So as I said, plasma membrane is the outermost layer of the cell. You can see here, this one is the outermost layer of a cell. One second. Okay. So this will be your outermost layer. This is one. This one will be called as the plasma membrane. Okay, you can see here. Then the inside material will be called as cytoplasm. This one. Okay, inside one cytoplasm, and the middle one, this one will be your nucleus. Okay, this one. See. Nucleus consists of again nucleoplasm, nuclear. All these things we will be studying when we are studying in detail about the nucleus. Okay. So what is the function of plasma membrane? It is going to separate the external internal environment, and it is selectively permeable. What do you mean by selectively permeability? All those things we'll be studying. Okay, when we are studying separately. Then you are having cytoplasm. Cytoplasm again contains cytosol and organelles in it, and nucleus. Nucleus contains DNA. DNA in the form of chromosome, and these chromosomes will make hereditary units called as genes. This is a structure. You know the structure. Now, one by one, we will be studying about the different parts of this cell. Okay, you can see here you are having lysosome here. You can once you have a look here, you are having centrosome here, centriole, cilium, flagella, microvillus is there. Okay, this is the microvillus, and this is mitochondrion, and this is nucleus, and this is your Golgi complex, or you can say Golgi apparatus. Then you are having smooth endoplasmic reticulum, and you will be having this rough endoplasmic reticulum. This one is rough endoplasmic reticulum, and this one is smooth endoplasmic reticulum. And these are ribosomes. Ribosomes are also attached to RER. Okay, so these are the different parts of your cell. Now coming to the plasma membrane. Now, what is this plasma membrane? Plasma membrane usually consists of two layers of lipids. Okay, it is a lipid bilayer. In intermediate, we might have studied the sandwich model and all these things, right? So here we are not going to study all the models. We know the standard one. What is it? It consists of two layers of lipid. It is a lipid bilayer. Okay, so this lipid bilayer, these lipid molecules, they contain three types of components. Okay, phospholipids you have, cholesterol and glycolipids. These lipid molecules are made up of phospholipids. Phospholipids means lipids attached to the phosphate group. Okay, phospholipids, cholesterol and glycolipids. Phospholipids will be constituting 75% of the total lipid molecules, and cholesterol will be constituting 20%, and glycolipids will be of 5%. Glycolipid means these lipids, they contain sugar on their sugar will be attached to the lipids, and these glycolipids will actually serve as a signature of a cell. Signature of a cell means we have a signature, right? And our signature will be unique. No other person will have same signature like us. In the same way, here also, these glycolipids, they serve as a recognizing components, recognizing structures of a cell. Okay, they serve as a signature for a cell. Now, as the most of the these lipid molecules, they are phospholipids. We will see the structure of a phospholipid, how it will. Okay. So, do you just imagine a matchstick? Everyone have seen matchbox, right? So, how a matchstick will be there? There will be one tail and there will be one head, round head. Black one is head, and lower one is tail. The same way the phospholipids have a head and two tails. Okay, so here the head will be hydrophilic in nature. Hydrophilic means it is a water loving group. Okay, so it is water soluble, hydrophilic, and this head will be attached to phosphate. It is a phosphate containing group, and this head will be facing outwards. Okay, how these will be attached to phospholipids? Tail, tail, this side head and this side head. Head, head. Okay, head, tail, head, tail. So the outermost one is your head, and the innermost one is also your head. In the middle, there will be this one. This, my fingers. These are your tails, and these are heads. My thumb is your head. Okay, heads and tails. Like this, they will be attached. Okay, two layers, lipid bilayer, one layer, external, internal. Okay. Now, so this is a tail, which is tail will be lipophilic in nature. Okay, it will be lipophilic in nature, means it is lipid soluble. Now coming to the functions of a plasma membrane. The main functions of a plasma membrane is to protect a cell from toxins, protect a cell from damage. Okay, protection. It serves as a barrier for toxins to enter into the cell, for drugs to enter into the cell. Okay, for foreign material to enter into the cell. It maintains the proper shape of a cell. Plasma membrane only gives a proper shape to the cell. It helps in cell junction as it contains transmembrane proteins. We will see that in next slide. It is going to help in the junction of a cell. Two cells will meet. It helps in the movement of a cell. And it is selectively permeable. Selectively permeable means what? It is going to allow only few compounds to pass through it. Okay, selectively permeable. That is very important function of the plasma membrane. If it is not selective, what will happen? Every material, anything which enters, if it can enter into the cell, then it will be harmful to the cell, right? So how it will be hazardous, right? So in that case, it selectively allows into the cell, allows into the cell. Okay, it selectively allows the compound into the cell. It is also going to help you in impulse transmission, where the signals will be uh, transduced from your ligand receptor, and how there will be impulse transmission, that we will see in next slide. Okay, future slides. Now coming to the selective permeability. So here there will be three kinds of permeability: high, moderate, and impermeable. Okay, high, moderate, impermeable. High uh, means it doesn't allow it doesn't need any permission to enter into the cell. Okay, highly permeable. For example, you want to join in a top college. Okay, the top college, will it allow every student to come? No, right? So it will select the students based on their merit, right? In the same way, here also our cell will select some compounds to enter. In that, the most topper students will definitely find, they will enter into the cell, right? In the same way, here also, the compounds like oxygen, carbon dioxide, and steroids, which are highly lipophilic in nature, they will enter into the cell without any permission. They are non-polar in nature, and they enter into the cell. They are highly permeable. The cell membrane is highly permeable to these kind of compounds. Okay, you are having certain other compounds like water and urea, which are small and uncharged particles. Okay, small and uncharged. Okay, now small and uncharged means, okay, they are average students. They will as they are scoring average. Okay, few seats are left. Now average students also they are taking moderate. Next is your impermeable. Impermeable means, now the students who did not get any rank, what they will do? They will go with the reference. Okay, or else what they will do? They will go for the management quota, right? In the same way, here also the glucose is impermeable. It cannot enter the cell without the uh, permission of cell membrane. So the cell membrane will never allow this glucose to pass. Why? Because first is, it is a large one, large uncharged polar molecules. Okay, large one, and ions, they will not be permeable. Okay, so because of its complexity, because of its uh, charged ones, uncharged ones, large structure, this glucose will be impermeable to cell membrane. Okay, so these are the different kinds of permeability. Now you will think that, okay, why we need to remember this? But you have to remember this because again, when you are going for pharmacology, there you will be studying about the drugs, and the permeability of the drugs will be like this only. Lipophilic drugs will be easily observed. Okay, whereas hydrophilic drugs will not be observed. So that this knowledge we are applying there. Okay. Next, coming to the membrane proteins. Now we have seen uh, phospholipids, right? So along with the phospholipids, there are different kind of membrane proteins which are embedded in the plasma membrane. You can see here, this blue colored structures, all are membrane proteins. Okay, these are membrane proteins. So these membrane proteins in the plasma membrane are of three types. Okay, the first one is integral proteins, or they are also called as transmembrane proteins. Transmembrane proteins means, see, this is, as I told you, this is the structure of lipophospholipid, right? So the proteins will be like this, present in the membrane, transmembrane, across the membrane. It is called as transmembrane. Okay, so they are called as either integral proteins or transmembrane proteins. You can see these blue colored structures, which we can here we can see integral protein. This is an integral protein. Okay, then you are having peripheral proteins, means just above the surface. You can see this is the head of the phospholipids, right? So just above the surface, some will be floating. They will be called as peripheral proteins. Just on the surface, they will be present. Then you are having glycoproteins. See here, this one. So these proteins will be attached to a glucose, any sugar molecule. Okay, that will be called as glycoprotein. Is it clear till here, ma'am? Yes. Coming to the functions of a plasma membrane, very, very important. It can be given for 5 marks, it can be given for 2 marks, or it can be given for 10 marks also. If it is given for 10 marks, then you are going to write the structure as well as the functions. If you are giving for 5 marks, then you are mentioning the uh, about the cell membrane and you are writing the functions in detail. Okay, and if it is for 2 marks, then you can just write the side headings, like in one line you can write each function, one line you can write. Okay, now coming to the functions, the first one is it is going to help in the transport of molecules, transport of ions, transport of glucose. I told you now, cell membrane is impermeable to glucose. Then how glucose is coming inside? By reference. Who is giving reference here? Our membrane proteins. They are opening the gates. Come, come, go from here. Okay, ions, they are also not permeable. So ions, they will be different ion channels. Ion channels are nothing but your proteins only, transmembrane proteins. Okay, so these channels are also proteins. So ion channels will open and allow the entry of ions. Okay, so they are going to help in the transport of ions. They are going to act as enzymes in different metabolic pathways. They are going to catalyze different metabolic pathways of a cell. Okay. Third function is they are going to help in the signal transduction. Signal transduction means if I say something, are you able to hear? Yes, right. You are able to hear. In the same way, here the cells will communicate with one another. Okay. So if I say something, what you should have to hear? You should have ears, and you should also have this Zoom link to hear, right? Zoom link or YouTube link, you should have that signal, right? If you don't have that signal, can you hear me? No. Why? Because you are having that signal before you are because you are having that link, you have clicked on it and you are hearing to my lecture, right? In the same way, the cells will communicate with one another. And how they are going to communicate? That is in by signal transduction pathways, which you're going to anyway see in the class. Then cell to cell recognition. Though these cells, they are going to help in recognizing each cell by they serve as markers for a cell. Okay, these membrane proteins sometimes they serve as a cell marker. I told you, glycoproteins. They serve as the signature of a cell. Signature means what? It is a marker of a cell. Okay. Intercellular joining. Some transmembrane proteins are involved in forming junctions. So the two cells will be joined with each other to transport substances between them. That is called as cellular junction. Some proteins will be involved in forming a junction, intercellular joining. And some proteins, they are going to help in the attachment of cytoskeleton, microtubules to give a proper shape to a cell. So these are the different functions of your membrane proteins. Transport, enzymatic activity, signal transduction, cell-to-cell recognition, intercellular joining, or we can say forming cell junctions and attachment. Next, coming to the cytoplasm. We have finished the plasma membrane. Now we are going to see about the cytoplasm. So this cytoplasm, the inner portion which we have seen in the diagram, it is called a cytoplasm, and it mainly it is a uh, part between the plasma membrane and nucleus. Okay, shall we see the structure once? So this is a plasma membrane and this is the nucleus. So whatever is in between, that comes under cytoplasm. And now cytoplasm contains two main components. One is cytosol and other one is organelles. Cytosols means whatever the fluid portion is there, that is called as cytosol. Okay. And this cytosol will be about 55% of total cell volume. Okay, and other constituents which are present in the cytosol. Okay, in the cytosol, fluid portion is 55%, and other constituents like water, ions, amino acids, proteins, lipids, ATP, glucose, fatty acids, and base products will also be present in the cytosol. Now coming to the cytoskeleton. Cytoskeleton, one second. Okay. So cytoskeleton means in the cell, inside the cell, there are certain filaments which will be extending throughout a cell. Okay, and those filaments are actually responsible to maintain a proper structure of a cell, and that is called as cytoskeleton. And uh, this cytoskeleton is made up of three types of filaments. You have microfilaments, intermediate filaments, and microtubules. Okay, microfilaments, these are the very thin, you can see here. See, this is the cell, and you are seeing some thin layers, thin tube-like structures, filaments which are coming, and they are going to give a proper structure to a cell. Okay. So first is microfilaments. In the microfilaments, these are the very thinnest elements, very like they are very thin. Okay, thinnest elements composed of actin, and most they are made up of proteins such as actin and myosin. And these microfilaments are usually present on the edges of a cell, so that they give a proper and mechanical support to a cell, and also they help in the movement of a cell. Now coming to the intermediate filaments, these intermediate filaments are somewhat thicker than microfilaments, but they are thinner than microtubules, and they are composed of several different types of proteins, and they are mainly present in the areas where continuously the cells are subjected to mechanical stress, such as your knees, your elbows, okay, shoulders. So wherever there is a continuous mechanical stress, there you will find these intermediate filaments in those type of cells. Coming to the microtubules, we are having large unbranched hollow tubes. Okay, just like pipe-like structures, hollow tubes. Internally, there will be hollow, composed of mainly tubulin. The protein here is tubulin. The protein in the microfilaments is actin and myosin. In intermediate filaments, they are made up of many proteins. Okay, so in microfilaments, actin and myosin. In microtubules, it is tubulin. Okay, say these are helping in determining the cell shape, and they also function in the movement of organelles such as secretory vesicles or chromosomes during cell division. Means during cell division they are helpful. They are going to give a specialized structures or cell projections to a cell such as cilia and flagella and all. Okay. See here, this is a microfilament. See, these are present on the edges, and they are mostly made up of actin and myosin. Intermediate filaments. They are present in the areas where there is continuous mechanical stress. Intermediate filaments. Okay, and these are microtubules. You can see here. They are going to help in the cell division. Okay, am I clear? Coming to the organelles of cytoplasm. So, what are the organelles present? So in the cytoplasm, along with the cytosol, there are different structures with characteristic shape, specific function in cellular growth and maintenance and in reproduction. They are having all the organelles have their own function, specific function. Okay. Now coming to the first organ, we are going to discuss is your centrosome. Okay. So centrosome here nothing but it is a center for microtubules organization. Okay, center for the microtubule organization, and it is located near the nucleus. Why? Because these microtubules are involved in cell division, right? So this is present near the nucleus, and this centrosome is mainly consisting of two parts. One is centriole, the other one is uh, pericentriolar matrix. Okay, centriole. This one is centriole. Okay, and the outer one layer will be this is pericentricular. Surrounding area will be pericentricular matrix. Okay, and these are microtubules. Can you see? If you see the TS means this is a pipe. Okay, you are cutting the pipe and you are observing from upwards. Okay, you are observing from upwards. So how you will see this is there will be cylindrical structures, each composed of 9 clusters of 3 microtubules. One second. Huh? So there will be a centriole. Okay. In the middle, there will be a centriole. Okay. And centrioles, they are cylindrical structures, each composed of 9 clusters of 3 microtubules. 9 clusters of 3 microtubules means you remove 9. Okay. Cluster of 3 microtubules means one cluster will be made up of 3 microtubules. Like that, 9 clusters will be present. You can see here in the diagram 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. Okay. So this is one cluster. Okay. One cluster of 3 microtubules. Like that, how many microclusters are how many clusters are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. Right? So like this, if they will be arranged, and surrounding the centriole, there will be pericentriolar matrix, which is going to hundreds of ring-shaped complexes. It contains hundreds of ring-shaped complexes, which is composed of tubulin. Coming to the next cell organelle, that is cilia and flagella. Here, cilia will be motile projection on cell surface. Cilia, you can see here how they are present. Okay, the major component will be microtubules again. Microtubules means what protein it will be? Tubulin only. Okay. Now cilia will be short, numerous and hair-like projections, and it contains a core of microtubules with one pair in the center. Just now how you have seen the centrosome, in the same way here there will be doublet microtubules. Doublet microtubules means you have seen 3 clusters. In one cluster, 3 microtubules, right? In the same way, cilia will be having cluster of 2. Okay, doublet microtubules you call it as. Like that, 9 clusters will be arranged in a circle, and middle there will be one doublet microtubule. Okay, middle one doublet microtubule surrounded by 9 clusters of doublet microtubules. Okay, next, this cilia is going to give you ore-like pattern of beating. Ore-like pattern means you have seen like the person who are going to in mostly in Kerala, we are using this in Onam. They will be showing in the movies also. They will show ore, how they will just move the water with a hand with a oar, and then they will be moving forward. Right? In that, what they are doing, they are pushing the water backward. Okay. In the same way, here also, cilia will be moving the fluid backward, and the organism will be moving front. Okay, so here you will be finding oar-like pattern of beating. Next, you are having flagella. Flagella are similar in structure, but they are somewhat longer than cilia. In cilia, there will be numerous. In flagella, only one or two will be present. Okay, and here flagella will be giving you wave-like motion, not oar-like motion. Okay, so it will beat, and so that the organism will move forward, but not it is not going to move the fluid around it. It is going to move the organism. So you can see here, cilia moves the fluid along the cell surface, whereas flagella moves entire cell. Next, coming to the ribosomes, you know about the ribosomes because they are the main structures which are responsible for protein synthesis. They are tiny granules which are composed of RNA and protein, and they are mainly consisting of two subunits. They are large subunit and the small subunit. You can see in the diagram here how they are present. And there are different type of ribosomes, but in human cell, that which type of ribosome is present now? 70S or 80S or 100S? 100S. Yes, ma? 80S. 80S ribosome will be present, and the larger subunit will be of 60S, and the smaller subunit will be 40S. Okay, 60S, 40S. Together they make up 80S. Okay, and they are made separately in the nucleolus. They are means larger subunit and smaller subunit are made separately in the nucleolus. Then they exit the nucleus, and they fuse together in the cytoplasm, not in the nucleus. Okay, they are where they are found. They are found in the rough endoplasmic reticulum, and they are found on the outer surface of the nuclear envelope. The main function of ribosomes is protein synthesis. Coming to the endoplasmic reticulum, in the endoplasmic reticulum, it is a network of membranes in the form of flattened stacks. You can see here, flat stacks, bags. Stacks means nothing but bags. Okay, just like balloon, if you widen the balloon, how it will be the same. Okay, flattened sacs or tubules. Okay, they are coming from the nuclear membrane and project throughout the cytoplasm. If you remember the diagram, you can know that. So there are usually two forms of endoplasmic reticulum, rough and smooth. Rough will be embedded with the ribosomes, and hence they are going to help in the protein secretion, whereas smooth endoplasmic reticulum, they are going to help in the synthesis of fatty acids, steroids such as estrogen and testosterone. Coming to the functions, this rough endoplasmic reticulum, as I told you, they are going to help in the production of proteins. They synthesize glycoproteins and phospholipids. Then from there, these phospholipids and glycoproteins are transferred into organelles. Then they are inserted in the plasma membrane, and whenever there is exocytosis, release of material constituents outside the cell, at that time they are secreted outside. Okay, smooth endoplasmic reticulum, as I said, they are going to help in the synthesis of fatty acids and steroids, and they are going to also help in inactivation of different drugs and other potential harmful substances. Coming to the Golgi complex are also same sac-like membranes. They are folded membranous flat. You can see here. Okay, flat membranous sac. It is present in all cells, but in large numbers, they are present in the cell that synthesizes and export proteins. The cells which synthesizes more proteins and export proteins, in those cells we are going to find more number of Golgi complex. The proteins from rough endoplasmic reticulum, they are going to move into the Golgi complex, where the proteins are packaged into a membrane-bound vesicle called as secretory granules. Now, these vesicles are stored in the cytoplasm, and whenever they are needed, they are moving to the plasma membrane, and from there the proteins are exported. Coming to the lysosomes. Lysosomes are membrane enclosed vesicles, means they are surrounded by a membrane. enclosed vesicles, just like a ball. Okay, they are called as they are formed from Golgi complex. Okay, so whatever the packing and all that is done in the Golgi complex. These lysosomes, they contain more than 60 kinds of enzymes in it, which are going to help in digestion and hydrolysis. Okay, so here whatever the microorganisms or microbes which are coming inside the cell, they are digested. Toxins, harmful toxins, all they will be digested inside the lysosome because it contains different digestive and very powerful digestive and hydrolytic enzymes. Okay, and also these lysosomal enzyme, they are going to help in recycle worn-out structures. Obviously, cell means definitely some structures will be worn out and new structures have to be produced, right? So what will happen to the worn out structures? Is there any what a dustbin in our body? No, no. So where they are going? So what happens to them is whatever the worn out structure is there, that will be engulfed by the lysosome. The lysosome will come and eat it. Okay, and then as it contains enzymes, it will digest it and it will return the digested components to the cytosol for reuse. This is called as autophagy, eating our own cell. Okay, next is mitochondria. So mitochondria, they are called as powerhouses of a cell as they generate most of the ATP through aerobic respiration. Okay, so usually they are located within the aerobic respiration means in the presence of oxygen. Okay, these are usually located within the cell where oxygen enters the cell or where the ATP is used. Wherever there is a requirement of energy, there you will find more number of mitochondria because there they are going to use oxygen, they are going to use the ATP and give you energy. So, coming to the structure of your mitochondria, you can see here, this mitochondria contains the external mitochondrial membrane and internal mitochondrial membrane. So, this one is external, internal, and there will be space between both of them that will be called as intermembrane space, and that will be filled with the fluid. Okay, and if you see the internal membrane is not smooth, but it is having different folds, and those folds are called as cristae. And inside there is a blue colored matrix that is called as mitochondrial matrix. And so whatever the enzymes which are responsible, which are present in the mitochondria, which are responsible for the cellular respiration, they are present in the cristae and in the mitochondrial matrix. Okay, this mitochondria also play a major role in apoptosis, where it is going to send some signals, that it is going to release some proteins which are called as apoptotic proteins. This is in future you will be studying how these mitochondria are helpful in cancer treatment. Okay, coming to the nucleus. Oval shaped structure which is present in the center of a cell will be called as nucleus. This nucleus is present in almost all the cells except matured RBC. In matured RBC, it is matured RBC is enucleated. Okay, so all the cells contain a single nuclei, but as your skeletal muscle is there, you will find several nuclei and skeletal muscle. That's it. In other cells, all the cells, single nuclei will be present. So this nucleus is covered by a double membrane called as nuclear envelope, which is similar to plasma membrane. And there are certain small openings in the nuclear envelope that is called, they are called as nuclear pores. Okay, and nuclear pore is nothing but a circular arrangement of proteins surrounding a large central opening. They are going to, nuclear pore is going to control the movement between the nucleus and the cytoplasm. Okay, and inside the nucleus you will find different spherical bodies called as nucleoli. This nucleoli is going to help in the production of ribosomes. Okay, and in the nucleus you will find genes which are the hereditary units and they are arranged along the chromosomes. And usually human somatic body, it contains 46 chromosomes, 23 from each parent. Now what is this chromosome? It is a molecule of DNA that is coiled together with several proteins This complex of DNA and RNA protein is called as chromatin. Okay, and the total genetic information which is placed on DNA is called as genome. This is the nucleus you can see here. It contains ribosomes, nucleoli, nucleoplasm. Inside this, whatever the fluid is present, that is called as nucleoplasm. The envelope, outer membrane will be called as nuclear envelope. Okay, and chromatin will be present, nucleolus will be present, nuclear pores will be present, small pores you can see here. Now coming to transport across the plasma membrane. How a substance is going to move through the plasma membrane. This is very important. Okay. So generally, you will be having two types of transport in the nuclear through the nuclear membrane. Sorry, through the plasma membrane. So passive transport and active transport. Okay. Passive transport means there is no requirement of energy. Energy is not required, means ATP is not used. It is based on the concentration gradient or electrochemical gradient. The ions will be moving. Okay. Next is active transport, where you require energy, where ATP is utilized. So you can classify the transport mechanisms through passive transport and active transport. In passive, you have osmosis, diffusion, and facilitated transport. And in active transport, you have having vesicles and protein forms. Okay. In vesicles, you are again having endocytosis, exocytosis, and pinocytosis also. Coming to the passive transport. So passive transport is the movement of molecules through the membrane in which there is no energy required. Okay. And substance will move downhill. Downhill means what? From its down its concentration gradient, means from higher concentration to lower concentration. Did you ever mixed ujala in the water? How it will be? First, when you not ujala, even if you go for any science experiments in your chemistry labs, maybe you have added some color, a reagent into a liquid. So first the color will be concentrated in the middle. Then it will pass through whole the liquid, and finally it will give you an equilibrium color to a liquid, right? So that is nothing but diffusion. Okay. Substance moving from higher concentration to lower concentration using its own kinetic energy without input of energy from any cell. Okay, that is called as diffusion. So here you can see movement of solute from an area of higher concentration to an area of lower concentration until equilibrium is attained. So when it will stop? When there is equilibrium, when it is distributed uniformly. So here it is. You have added the color. You can see it is settled down here. Here high concentration and here it is low concentration. So it is moving from high concentration to low concentration and finally equilibrium is attained. That is your diffusion. And the factors on which the diffusion depends is steepness of the concentration gradient, temperature. More steepness, more diffusion. More temperature, more diffusion. Mass of the diffusing substance. Mass of the diffusing substance. Just imagine a person who is fat and if he wants to move, he will take some time, right? Even if he want to come from the hill, he will be slower when compared to a person who is very thin. Okay. So higher mass substance, low diffusion. Lower mass substance, high diffusion. Okay. And the surface area, more surface area, more diffusion. And diffusion distance also, less distance, more diffusion. Coming to simple diffusion, as I said you, this is a simple diffusion where there is not, means in this diffusion, there is no, this molecule is not taking help of any channel or any carrier. Okay. So it is a passive process in which this substance move freely through the lipid bilayer. It is not going to ask any help. Okay. From the membrane proteins. Okay. So non-polar hydrophobic molecules. This is very important. Non-polar hydrophobic molecules like oxygen, carbon dioxide, fatty acids, steroids, fat soluble vitamins like ADEK, they easily pass through the membrane. Okay. Whereas small uncharged polar molecules. Small uncharged and polar molecules like water, urea, and small alcohols, they need some help from the membrane proteins. They alone can't go. They have to take some help from the membrane proteins. They always require some help from their friends. Okay. So coming to the facilitated diffusion, this facilitated diffusion, as I said you, simple diffusion, they do not require any kind of help. Okay. Whereas facilitated diffusion means your friend is holding your hand and taking you along inside. Okay. Simple diffusion means directly you are going outside, you are coming inside. You are going outside, you are coming inside. You don't require any friend to come with you. But here you require your friend to hold your hand and pull inside. Okay, that is called as facilitated diffusion. But here there is no energy required. It is not like pulling, but out of love, your friend is just opening the door for you. Okay, so he is not using any energy. So coming to the facilitated diffusion, substances that are too polar, which are highly polar. Here, these small uncharged polar molecules also they undergo simple diffusion only. Why they undergo simple diffusion is because they are small in nature. The size of the substance is small, so they can easily pass through the membrane. But here facilitated diffusion, the substances which are too polar or which are highly charged, they cannot move through the layer, lipid bilayer. So therefore, they require certain carriers or channels. If a ion is utilizing a channel, then it is called as channel mediated facilitated diffusion. You can see here. If it is through channel, it is channel mediated facilitated diffusion. If it is through carrier, it is carrier mediated facilitated diffusion. Okay. And this is simple diffusion. No one is required for it. Okay. Now, channel mediated diffusion. In this, what happens is a solute moves down its concentration gradient through a membrane channel or an ion channel. So a membrane or an ion channel. So these ion channels are specific for specific persons. Already, obviously, right? Your friends will be specific to you. It is not like one friend will be friend to everyone. Everyone will be having one best friend, right? In the same way, the ion channel will be specific to specific ions. Likewise, you have potassium channels, sodium channels. Potassium channels will not allow sodium ions to come inside. Okay, and sodium channels will not allow potassium ions to come inside. Now, next, carrier mediated facilitated diffusion. Here, instead of channels, if you say channels will be present for ions, but for some molecules there will be no channels. Like for example, glucose you are having. Glucose will not have any channel. Did you ever heard the glucose channel? No. So, so how they will go inside? Now she will, glucose will be thinking how I should go, I should go, I should go. Now he will think of an idea. Okay, I will sit on a carrier and I will go inside. Okay, so anything in this mechanism, what is happening is a carrier or a transporter. It is going to move a solute down its concentration gradient. The solute binds to a specific carrier on one side and released on the other side of the membrane. For example, you see here. Now, this structure of glucose will be this structure of glucose. Okay, sorry. This structure of your carrier, it will like glucose. So it will allow the glucose to sit on it. Once it sits, it will change the structure. Okay, it will change the structure. It will be like this. It will take glucose. Then after taking glucose, it will become like this. Okay, when it becomes like this, it will leave the glucose inside the cell. In this way, how this glucose is going to get carried inside by a carrier. Coming to the osmosis. Here, it is a type of diffusion which involves a movement of solvent, where in the diffusion we have seen the movement of solute. Here we will see the movement of solvent through a selectively permeable membrane from higher solvent concentration to lower solvent concentration. Don't get confused because might have studied in your intermediate like osmosis is low pressure to high pressure, madam. Yeah. So here we are talking about the solvent. We are not talking about the solute. Okay. Solvent movement from higher solvent concentration to lower solvent concentration. Okay. Higher to lower concentration through a selectively permeable membrane. That is called as osmosis. So during osmosis, molecules can move across the membrane by moving between the neighboring phospholipid molecules to the plasma membrane, or they can move by aquaporins, which are small pores present in the plasma membrane. Okay, so this this process osmosis will occur only if the membrane is permeable to water but not permeable to the solute. The membrane, the selectively permeable membrane should be permeable to water but not to solutes. It should be selectively permeable. It should not be permeable to any kind of solute. Okay. Where you have seen the selectively permeability? Can anyone say? Can anyone say? In kidneys, you will see. Okay, selectively permeable in nature. When you study the structure of nephrons, you will see this. So you can demonstrate this principle of osmosis by using a small experiment where you are taking a U-shaped tube and you are filling it with water and some type of solute, and in the middle you are placing selectively permeable membrane. The solute is present in right arm but not in the left arm. Okay, so you are see here. So what is here? Here there is higher concentration of water. I'm not talking about the solute. Okay, higher concentration of water and low concentration of water here. Okay, so what happens here? So when you are leaving this for a certain period of time, the movement of molecules will be moving from your left arm to right arm, so that the water is balanced. Okay, so that the water is balanced. Concentration of water is balanced. Next, if you want to restore the conditions, so how you will restore? If you simply put pressure on the left arm, the water will come back to, sorry, if you put simply the pressure on the right arm, the water will come back to the left arm. Okay, so this is osmosis. How the movement of the molecules or water is from left arm to right arm. This is the principle of osmosis. And how much pressure you are applying to get back the original state, that is called as osmotic pressure. That is called as osmotic pressure. Now, where this osmotic pressure concept is used is, you can see here, when you take an RBC, okay, and you are putting it in some solution, okay, in our blood cells also, there should be what the measurement of this osmotic pressure, that will come under tonicity. Okay, so you might be hearing about the saline solution. That is, what is saline solution? It is 5% normal saline NaCl solution, right? So here, if you take an isotonic solution, okay, isotonic solution means which is having same osmotic pressure as that of our blood. Okay, so if you're taking a normal saline, then and you put that RBC in the normal saline isotonic solution, then the RBC will be normal in shape. It will not get destroyed. Okay, whereas if you put the RBC in the hypotonic solution, okay, hypotonic solution, then what will happen? There will be movement of water molecules from higher concentration, higher solvent concentration. I am saying hypotonic. Hypotonic means the solvent is less in the water, in the solution which we are taking, and the concentration of water is more in the RBC. Okay, so what happens is, one second. So what happens is, the water will be moving from higher concentration to lower concentration, and all the water will enter into the RBC. The RBC will swell and finally it will break down. That will be called as hemolysis. And if you put the RBC in the hypertonic means higher concentration of water is there in the hypertonic. Okay, so if you are putting this in hypertonic solution, sorry, here hypertonic solution means the concentration of solute will be more in the solution. Okay, so what happens when you put the RBC in this hypertonic solution is, all the fluid from the RBC will come out. Okay, and there will be crenation. Crenation means the RBC will shrink. Okay, and if you put in hypotonic solution, all the water from your solution will enter into the RBC and leads to swelling of RBC, leading to hemolysis. Hypertonic solution, crenation. Very, very important in all your competitive examinations. Coming to the active transport. Active transport will be called as uphill transport, and it utilizes two sources of cellular energy. Okay, ATP and energy stored in ionic concentration. Okay, here ATP will be used. ATP will be used to move the molecules around it. Okay, so that will be called as active process. Uphill transport means here the ions will be moving from lower concentration to higher concentration. Uphill, you are moving up. Definitely, when you are moving up, there should be some energy required, right? So that is why it is called as uphill transport. Now you are having two types of active transport systems: primary active transport and secondary active transport. Okay, so primary active transport means, the best example for primary active transport is Na plus K plus ATPase. Okay, it is a pump which is generally present. This Na plus K plus ATPase is mainly used in maintaining the equilibrium of a cell. Okay, so how this pump works? First, I will say what is the use of this pump. Okay, so you know the structure of a cell, you know action potential, right? When there is stimulus, there will be influx of sodium ions and outflux of potassium ions. Okay, when there is a stimulus, there will be impulse generated, there will be influx of sodium ion, outflux of potassium ions. Why that will happen? Why? Because the sodium is more outside, it is coming inside, and the concentration of potassium is more inside, it is going outside. When the doors are open, obviously, if you see, if you are having a small two rooms, okay, the concentration of people are more in one room and it is less in another room. Immediately, when you open the door, what happens? Automatically, the persons will start moving to another room. In the same way, the sodium ion concentration will be more in the extracellular fluid and less in the intracellular fluid. So whenever you open the door, the sodium ions will be influxed. Okay. In the same way, the potassium will be more inside. Whenever you open the door, immediately they will run away from the cell. That is potassium ions. Okay. So now this Na plus K plus ATPase is going to maintain that concentration. It will always see that there is more sodium outside or more potassium inside. When there is action potential, there will be influx of sodium and outflux of potassium, right? So after that, there should be balance. Again, when there is the next impulse, again this movement should occur, right? So how that concentration is maintained? That is because of Na plus K plus ATP is pump. It is going to pump the ions, that is why it is called as pump. So how it is going to work? So first, what it should do? It should maintain high concentration of sodium outside the cell and high concentration of potassium inside the cell. Okay, so whenever there is an impulse generated, there should be influx of sodium and outflux of potassium, right? So it has to bring sodium from outside and it has to leave the potassium outside. Understood? It has to bring sodium from outside and leave potassium from inside to outside. Okay, so first the transmembrane protein, you can see here the structure will be like this. Okay, now this structure will facilitate the binding of sodium ions. You can see here 3 sodium ions are going and binding to the protein or channel. Okay, now as soon as the sodium binds to the channel, it changes its shape because it is a protein. Whenever there is any change in the structure, it will change its own structure. Okay, so it will change its shape and it will become like this. It was like this, now it will become like this. So when it is like this, immediately this structure will not favor sodium ions to get attached. So the sodium ions they will leave the proteins. Okay, they will leave the channel. So now sodium ions came inside. Now this structure, whichever it is like this is going to facilitate or they are going to attract potassium ions, 2 potassium ions. So these two will come and bind. Immediately when they come and bind, again it changes its shape to like this. So immediately the 2 potassium will be sent inside. So like this Na plus K plus ATPase will require, it will actually, when this happens, when this sodium is binding to the protein, there will be hydrolysis of ATP to ADP and phosphate. Phosphate will be attached to the protein or the channel. Okay, it will be attached to the channel and this will make the channel to change its shape. Okay, so in this way ATP is utilized and Na plus K plus ATPase is working to maintain the concentration of sodium and potassium in the cell by utilizing primary active transport system. Next is secondary active transport. Here, the secondary active transport means they are going to use the energy of ATP hydrolysis, but they are not going to use it directly. They are using it indirectly. So what happens here is whenever one channel is opening, immediately the other ions will take advantage of the previous channel and they get along it. For example, if you see here, there are 2 types of channels here, antiporters and symporters. Antiporters means, now if you have seen in the active transport, they are getting sodium inside, one channel is getting sodium inside, the same channel is one channel is the same channel is sending the sodium outside, the same channel is getting the potassium inside. In the same way, here what happens is there will be movement of 2 ions. Okay, if the 2 ions are moving in the same direction, okay, along with the sodium, if glucose is also coming inside, okay, then it is called as symporter. And if the movement of ions is in different direction, one is coming inside and the other is going outside, then it is called as antiporter. Antiporter. You can see here that they are going to use the energy which is present in sodium Na plus or H plus concentration gradient. This energy is used to drive the other substance across the membrane against their own concentration gradient. So what happens here is, if you see an example of this, sodium and calcium. Okay, usually Na plus H plus this concentration gradient will be there. Okay, they will utilize the ATP. So when these channels are open, okay, when this channel is open, automatically these channels will also get opened. Okay, so what this calcium will do? Okay, sodium is coming inside. No, the door is opening. I will go out. I will go out. It will go out. H plus will go out. Okay, H plus will go out and calcium will also go out. Okay, so here what is happening? Sodium is coming inside, calcium is leaving. So it is antipolar. Calcium, sodium is coming inside and H plus is leaving. So it is antipolar. Okay, in the same way, if you see here, sodium and glucose. When the sodium is coming inside, along with it, glucose is also coming inside. Okay, with the extracellular fluid. So that is called as symporter. So if any other ion is just taking a chance and coming inside along with the sodium, then it is called as secondary active transport. It is not using the energy directly, but it is using it indirectly. Next, coming to the transport in vesicles. So now we have seen how molecule is transporting through diffusion, osmosis, and through active transport systems, right? Now there is one more transport system which occurs in vesicles. So what is a vesicle? Vesicle is a small bag-like structure or spherical sac-like structure which is going to cover the subject, cover the substance and move it along the cell. Okay. So if a substance is moving into the cell, from outside it is coming inside. If a substance is moving into the cell, it is called as endocytosis. And if a substance is moving outside the cell, then it is called as exocytosis. Okay. Endocytosis is again classified into exocytosis. You will not be studying here because it is a process usually you will see in the neurotransmission thing and you need not study it. We are going to concentrate on endocytosis. Endocytosis is classified as receptor mediated endocytosis, phagocytosis and pinocytosis. Okay. So this is receptor mediated endocytosis. The best example for this is LDL receptor complex. So here the plasma membrane will be having certain receptors and the area where this is present is called as clathrin coated pit, and the LDL molecules whenever they find they come and bind to the receptor. Once they bind to the receptor, the plasma membrane will invaginate. It forms a cup-like structure. You can see here how it has invaginated. It will invaginate and take all the LDL particles, and this vesicle or this sac-like structure will get pinched off. It separates from the plasma membrane and it forms a vesicle. Okay. Now this vesicle is coated with this clathrin protein will be coated around the vesicle, and step 3 is this clathrin protein will leave it and then it is called as uncoating. Okay. After uncoating, this uncoated vesicle, it will fuse with transport vesicle. One more vesicle, it will fuse with the transport vesicle, and this transport vesicle is actually involved in recycling of receptors to the plasma membrane. So here see, receptor is also there and ligand is also there. LDL is also there and receptor means the one which is going to help in the binding of ligand to a cell. Okay. So that is also there. So receptors and LDL will be separated. Receptors move to the transport vesicle and sorry, LDL will be moving to the transport vesicle, and from here this transport vesicle will fuse with the lysosome. Lysosome contains, as I said you, there will be different digestive enzymes, and because of the digestive enzymes, this LDL will get into it will get degraded. It will be dissolved or digested. Okay, and there will be degradation in the lysosome. This is how a receptor mediated endocytosis will take place. Okay, first there will be receptors on the cell surface. LDL will come and bind to the receptors. There will be invagination of a plasma membrane, and it will get pinched off and it will form a sac. Now this sac is covered by clathrin protein that will be removed, that is called as uncoating. Then inside there is LDL attached to the receptor, so receptors should be reused. So for recycling purpose, these receptors are separated, LDL particles are separated. Separated receptors are again going back to the plasma membrane through transport vesicle. In another transport vesicle, there will be presence of LDL particles. This transport vesicle now it will fuse with the lysosome, and as lysosome contains different digestive enzymes, there will be degradation of LDL. Okay, this is receptor mediated endocytosis. Next is you are having phagocytosis. In the phagocytosis, what this is similar to that of endocytosis only, but the thing here is the cell will engulf the solid particles. Phagocytosis means what? Phago means khana. Khana means eating. Okay, cell eating you call it as. So usually when there is a bacteria or virus, our macrophages, neutrophils, they eat the bacteria. They engulf the particle and they digest that bacteria and virus inside them. The same is seen here. This is green colored one is a microbe, and when it comes and enters, the macrophages will invaginate. First, the plasma membrane of the macrophages will invaginate. It will take the microbes inside, then form a phagosome. Now it will not you will not call it as a vesicle here. You call it as a phagosome. Now this phagosome is going to combine with the lysosome, and lysosome as it contains digestive enzymes. This microbe will be digested and the residual body will be formed in the cell, which will be again eaten away by the lysosomes only. Now coming to the different topic, that is cell junctions. Cell junctions means how cells contact with each other. There are 5 different types of cell junctions. You have tight junctions, adherence junctions, desmosomes, hemidesmosomes, and gap junctions. Okay. So one by one we will see. Tight junctions means there will be very tight junctions. It will be like web-like. You can see in the picture how they are present. Okay, the web-like strands of transmembrane proteins. Transmembrane proteins will be very tightly fused to one another, and there will be no chance of any molecule going inside. Okay, they seal off the passageways between the adjacent cells. They inhibit the passage of substances. They will not allow the passage of substances between the cells, and they prevent the contents of these organs from leaking into the blood or surrounding tissues. Next you have is adherence junctions. Adherence junctions means there will be a dense layer of proteins inside the plasma membrane. This will be going to attach both the membrane proteins, okay, and to microfilaments of the cytoskeleton. Now transmembrane glycoproteins called cadherins join the cells. You can see cadherins here. These one. Okay, the middle one will be called as cadherins, and the cadherin inserts into the plate from the opposite side of the plasma membrane, partially crosses the intercellular space and connect the cadherins of the adjacent cell. So the cadherins are the proteins which are going to connect from one cell to the other cell. They cross like this, like this. Can you see him here? Okay, means they are covering, they are going deep inside the cell, more than half of the cell. Okay, that is called as cadherin. And whatever the tight strip which is formed, that is called as plaque. Plaque cadherins go inside intercellular space and they connect to the cadherins of the adjacent cells. Okay, so here they will these adherence junctions are going to help the epithelial surfaces resist separation, and during various contractile activities when the food is moving, they will not allow the layers to get separate out. Okay, that is called as your adherence junctions. Next you are having is desmosomes. Here the plaque and the cadherins are present, but they are not attached to the microfilaments, but they are attached to the cytoskeleton. They are not attached here, but they are attached on this side. You can see here on the sides. Okay, they are attached to intermediate filaments. They are not attached to the microfilaments, but they are attached to the intermediate filaments. Okay, and these are actually, they are going to give the structural stability to the cell. Desmosomes. Now here hemidesmosomes, here they just look like desmosomes. That is why they are called as hemidesmosomes, means half desmosomes. Okay, so here the glycoproteins, the transmembrane glycoproteins in hemidesmosomes are integrins rather than cadherins. Here the protein is different. There the protein was cadherin in desmosomes, but in hemidesmosomes the protein is integrin. Okay, and but these look like desmosomes. That is why they are called as hemidesmosomes. Okay. On the inside of the plasma membrane, the integrins attach to the intermediate filaments made of the protein keratin. Okay. So here, inside what they will be doing this, integrins attach it to intermediate filaments. These intermediate filaments, so these are integrins. They attach it to filaments such as keratin, which are made up of keratin. Okay. These are intermediate filaments, or I can say integrins and keratins. Okay, so they are usually present in the basement membrane. Okay, they are present in the basement membrane of the cell, where if you see in the epithelial cells, when you study in tissues, you will see basement membrane and the outer layer of cells. So they are usually present in the basement membrane, and thus hemidesmosomes, they will anchor the cell to the basement membrane, but they are not allowing both the cells to attach. Desmosomes, what they are doing, they are attaching the cells like this. Hemidesmosomes, what they are doing, this is one cell. Only this is, in these hemidesmosomes are going to help in attachment of the outer part of the cell to the basement membrane. Okay, so it is only inside cell. It is actually not a cell junction, but as it looks like desmosomes, it is coming in this topic. Okay, hemidesmosomes. The last one is your gap junctions, where gap junctions, as the name indicates, that there are large gaps between the cells. Okay. Now here, whatever the connections or the membrane proteins which are involved, they are called as connexins, means connecting. They are connecting each other, connexins. Okay. These connexins are, they are tiny fluid filled tunnels called connexons that connect neighboring cells. Okay. The plasma membrane of gas junctions are not fused together as in tight junction. The plasma membranes are not fused together as a tight junction, but they are separated by small gaps. Okay. So as there are many gaps, the fluid from one cell to another cell flow very easily. The communication is easy in the gap junction. Is there any communication in the tight junction? No. In desmosomes? No. In hemidesmosomes? No. In gap junctions? Yes. Next one is your cell signaling system. So cell signaling is a vast process, and here we will just discuss overview. Why? Because again, actually the thing is, you should just know the concept of cell signaling system, and the actual mechanism you will be studying in your pharmacology. Okay. So what is the cell signaling? It is a process by which one cell communicates with the other cell within the body or with the external environment. How it will communicate? One cell will be sending signal, that is through a messenger, that is your ligand. Okay. Now this ligand should go and attach to the other receptor. Okay. How it will attach? There should be some specific point. That is nothing but your receptor. What is receptor? It is nothing but a transmembrane protein which recognizes ligand and allows the ligand to bind to it, so that it will process further and give response. Okay. If there is no receptor, there will be no response from the cell. Okay. So usually there is a cell to cell signaling involves transmission of a signal from a sending cell to a receiving cell. Okay. So there are different signaling systems. So there are four types of signaling systems: paracrine, autocrine, endocrine, and signaling by direct contact. Okay. Paracrine means the signal from one cell is going to the other cell. Okay. Cells that are nearer to one another. I am saying something, I am sending some message to my person who is sitting next to behind to me. That is called as paracrine signaling. Okay. Autocrine signaling means here the cell has to release some uh, mediator which will act on it only. Autocrine signaling. Okay, this is usually seen in immune cells, immune system. Okay, so in autocrine signaling, a cell signals to itself, releasing a ligand that binds to receptor on its own surface. It will release a ligand which will come and bind to its own surface. Okay, for example, you are seeing many autoimmune diseases. Okay, rheumatoid arthritis. What it is that our body actually have to give antibodies to antigen. Okay, but there is no autoimmune disease. What happens is, without generally speaking, our body should give antibody. It should produce antibody to an antigen when there is an antigen. Okay, but in some cases, in this autoimmune disease, what happens is our body fails to recognize our own cell. Okay, they will fail to recognize our own cell. They think that our own cell is an antigen and they start producing antibodies. So where they are acting, the antibodies are acting on our own cell. So that is type of autocrine signaling. Okay, next is your endocrine signaling. Now what happens is, in paracrine I am speaking to the next person, so that person is able to hear. But in endocrine signaling, I want to speak to some other person who is sitting very far behind me. So what I will do, I will write a message. I will drop in the drop box, which is moving from here to there. Okay. And when it reaches to the other end, the person who is sitting far away from me will take and read the message. In the same way, endocrine, what is happening here is our glands are there. They release hormones in the blood. Blood will be carrying the hormones, and wherever they are required, they are acting. They are acting on that particular organ. Okay, thyroid gland is giving out T3 and T4. T3, T4 have many functions. They are actually acting on all your body parts, right? So how they release this in the blood? Blood will be moving all over your body surface. So wherever the receptors are there, they are going and binding to the receptors. That is your endocrine signaling. Okay. And signaling through direct contact, the best example what we have seen is gap junctions. Like directly they are sending the molecules from one cell to another cell by gap junctions. What we have discussed before, the same thing. Okay. Next. Now, stages of cell signaling. So, how a cell will signal? First is a sending cell will be giving one signal. That signal should go and bind to the receptor. When it binds to the receptor, there should be some transduction going on. Okay, where different chemical signals will activate, enzymes will activate, and finally we will get a response. So you are having different cell signaling pathways. Okay. Cell signaling pathways usually depend on what kind of ligand it is. Okay. For example, if you are having lipophilic ligand like steroid and vitamin D3, okay, they will, what did I say? If the molecule is lipophilic, it can cross the membrane very easily. Why? Because the tail part of your transmembrane protein is lipophilic in nature. So they both are friends and they will allow the lipophilic agents to pass through. If the ligand is hydrophilic, okay, if the ligand is hydrophilic in nature, what is ligand? Anything which binds to the receptor. Okay, so if the ligand is hydrophilic in nature, they cannot go inside. They cannot go inside. So they will stand on the plasma membrane. They will bind to the receptors which are present on the plasma membrane, and they will give different signals which will actually give different they will activate different pathways, and finally you will get a result. Understood? If a ligand is lipophilic, directly it will cross the plasma membrane and attaches to the receptor which is present inside the cell, intracellular receptor. But if a ligand is hydrophilic in nature, it cannot go through the plasma membrane. It has to attach to a receptor on the cell surface, that is extracellular receptor, and then this binding will cause many reactions, many pathways, and that will cause a response. Okay. So in this, you are having four different types of channels by which you are going to first three will be for hydrophilic ligands, and the last one nuclear receptors will be for lipophilic ligands. Okay, so first three ligand gated, G protein coupled receptor, and tyrosine kinase linked receptor. All these will be for hydrophilic compounds, hydrophilic ligands. Okay, so in ligand gated, I'll just explain the diagrams. You can see here. So in the ligand gated, what happens is whenever a ligand comes and binds to the ion channel, channel will open and it will allow the ions to move in. Okay. Whenever a ligand comes and binds to the receptor, the ion channel, receptor and ion channel. The ion channel itself is a receptor because everything other than phospholipids, whatever the structures we are studying in the plasma membrane will be receptors only. Proteins only, transmembrane proteins. Transmembrane proteins only behave as receptor, behave as ion channels, behave as transporters. Okay, behave as carriers. Okay, so here they come and bind to the ion channel. Whenever they bind, immediately the channels will open and they will facilitate the movement of ions either inside or outside depending upon the concentration gradient of a cell. Okay, next. Now G protein coupled receptors, it is very important. They are also called as 7 transmembrane. Why? Because why 7 transmembrane means you can see the basic structure of GPCR. So it is like this: 1, 2, 3, 4, 5, 6, 7. So 7 times it is coiling around the plasma membrane. That is why it is called as 7T or 7 transmembrane domain receptor. As it is in the form of a snake, it is also called as serpentine receptors. Okay. So now what happens is, so G protein is actually, it is a protein, one assigned one into a protein, and you can see how it is, and it will be having two terminals, N terminal and COOH terminal here, inside here, C terminal, you can see here. C terminal and N terminal. N terminal will be having amine group to it attached here, and this amine group will be facilitating the attachment of ligands. Okay, and C group, it will be attached to the G protein. Okay, this is G protein. G protein consists of alpha, beta and gamma subunit. Okay, this is the receptor, G protein coupled receptor. It is receptor, it is G protein. Together it is called as G protein coupled receptor. A receptor which is coupled with G protein. This G protein is nothing but alpha, beta and gamma subunits. It is made up of alpha, beta and gamma subunits. Understood? Now, so in this system, when the G protein is activated, there are several secondary messengers released in the cell. Those secondary messengers are, they are going to relay the signal, means they are going to send the signal, they are going to pass the information from the cell surface to the target molecule inside the cell. Okay, so what are the certain secondary messengers you have is diacylglycerol, DAG, phosphatidyl inositol, you have CAMP, CGMP, IP3, calcium, and gases like nitric oxide, carbon monoxide, and H2S. Okay, very important. Okay, what are the examples of secondary messengers in competitive exams, in 2 marks questions? Okay, and everywhere, wherever you find it, you can find this question of secondary messengers. So what are the secondary messengers doing? They are carrying the information from the cell surface receptor to the target molecule inside the cell. What are they? They are different molecules like DAG, diacylglycerol, phosphatidyl inositol, CAMP, CGMP, IP3, calcium, and gases like nitric oxide. Now, what are the signal molecules or ligands? Ligands means what? The one which is going to attach to the receptor. So it can be a drug, it can be photon, ion, amino acid, it can be odorant, it can be pheromone, it can be icosanoid, it can be a neurotransmitter, it can be a peptide, it can be protein, it can be a hormone. Okay. So now here the signal transduction in GPCR occurs by three pathways: CAMP pathway, IP3DAG pathway, and CGMP pathway. Okay. So CAMP pathway, what happens here is, this is the normal G protein coupled receptor coupled with G protein. Okay. Now ligand is coming and binding to the receptor. Now what is happening? This alpha subunit is getting separated. See, beta, this G protein first it got separated, beta, gamma separated, alpha separated. Alpha subunit will go and bind to the adenylyl cyclase. It is an enzyme which is present in the plasma membrane. Okay, it binds to it. So actually, what happens when the ligand is binding to the receptor is, the GDP which was bound to G protein will be converted to GTP. Okay. Now GTP will be there, and this will facilitate the alpha subunit of G protein to bind to adenylyl cyclase. So when this adenylyl cyclase is activated, it will convert ATP to CAMP, cyclic adenine monophosphate. Okay, cyclic adenosine monophosphate. So when it is happening, what happens? CAMP levels will increase. As the CAMP levels will increase, it is going and activating your enzyme such as protein kinase. Okay, protein kinase is an enzyme which contains two regulatory and two catalytic units. So these units get separated, and there will be activation of protein kinase enzymes. Now these protein kinase enzymes will be going and activating, and they are going to um, alter the protein synthesis in the cell, and thereby the cell function will be altered. Understood? The ligand will bind to the receptor. Receptor, before the ligand binds, it is bound to G protein. G protein is bound to GDP. Okay. Now, once a ligand comes and binds, G protein gets separated. GDP gets replaced, and alpha subunit will go and activate adenylyl cyclase. It is going to convert ATP to CAMP. As the CAMP levels increases, it is going to activate your protein kinase enzyme. Protein kinase enzyme will be going to alter the cell function. Okay. Next is your IP3DAG pathway. The same thing happens here. Instead of here adenylyl cyclase, the other enzyme will be activated, that is your phospholipase C. Okay. So when the phospholipase C is activated, it is going to hydrolyze phosphatidyl phosphate PIP2 into IP3 and DAG. PLC phospholipase C is going to hydrolyze PIP2 into IP3 and DAG. Okay, this IP3 is going to activate the calcium channels, and calcium ions are going to activate your PKC, protein kinase. Okay, protein kinase enzyme. Now here also C means catalytic units. Okay. Now DAG is also directly going to activate your protein kinase, and next there will be downstream like alteration of the cell proteins and cell function. That cell function will be altered. So this is the main pathway. Okay. Ligand will come and bind to the receptor. G protein gets separated. GDP will be replaced. Alpha gets separated, and it will go and bind to the phospholipase C. Phospholipase C is going to hydrolyze PIP2, which is present intracellularly. PIP2 is present on the plasma membrane. This is going to hydrolyze into IP3 and DAG. These two are secondary messengers. Now IP3 is going to activate the calcium channels. IP3 is going to increase the calcium levels by endoplasmic reticulum will actually be storing the calcium ions. So that calcium ions will be released inside the cell, and DAG will be going directly activating your protein kinase enzyme, and finally there will be change in the cell function. The last one is you are having CGMP pathway. So in the CGMP pathway, what happens is, you see from here, when there is nitric oxide is going to activate guanylyl cyclase, just like how adenylyl cyclase is activated, PIP2 is activated. In the same way here, guanylyl cyclase, GC, will be activated. This activated GC will be converting GMP to CGMP. As the CGMP levels increases, there will be vasodilation. Okay, just remember that thing, that this everything actually in detail you will be seeing in your pharmacology. Just remember what are the secondary messages which are involved and how the pathway, which enzyme is getting activated. That's it. Okay. Next, you are having is tyrosine kinase linked receptors. Here, these receptors, they are actually high affinity self-surface receptors. They are having high affinity to the cytokines and hormones, especially to the cytokines and the hormones. Okay. Now, tyrosine is an amino acid. What is kinase? Kinase is an enzyme which is going to transfer phosphate from ATP to specific target molecule. So suppose this is ATP and this is I am having substrate. I am putting some kinase. If I am putting some kinase here, this phosphate from ATP will come and attach it to the substrate. One phosphate from the ATP will come and attach to the substrate. That is called as kinase. Now, as this enzyme is specifically targeting tyrosine amino acid, it is called as tyrosine kinase linked receptors. Now, this is the basic structure of tyrosine kinase linked receptor. They are having extracellular domain. This is outer domain, and this is intracellular domain, and there will be alpha and beta units. The one which is chains, these are called as. Okay. So whenever a ligand comes and binds to it, they will form a dimer. Dimer means two receptors will come together. Okay. And there will be phosphorylation of the enzyme. Okay. There will be phosphorylation and there will be activation of cell. That one, in this, you are having two pathways, RAS-REF MAP kinase pathway and JAK-STAT pathway. Okay. In this, ligand comes and binds, then there will be formation of dimer. You can see the phosphate groups are attached to it. And here what happens is, all these RAS, GAP, RAF, MEC, MAP, all these are different types of proteins present in the cell. That's it. Okay. So you have to, you need not remember all these proteins. Again, this will be coming in the pharmacology. Just for your understanding, I am explaining you formation of dimer and there will be series of activation of different cells, different proteins, series of activation of different proteins, and finally there will be change in the cell function. In JAK-STAT pathway also the same way, the proteins here involved are JAK and STAT. That's it. Okay, the protein names are JAK and STAT. That is why it is called as JAK-STAT pathway. In detail, we'll study. And finally, you are having nuclear receptors. Okay, nuclear receptors. This we are going to talk about the ligands which are lipophilic in nature. What I said, lipophilic means they do not attach to the outer membrane. They directly come inside the cell and they get attached to the receptors which are present in the cell, in the cytoplasm or in the nucleus. Okay, in the cytoplasm or in the nucleus. If the receptor is present in the cytoplasm, it is called as type 1 nuclear receptor. And if the receptor is present in the nucleus, then it is called as type 2 nuclear receptor. You can see here, if it is an hormone, steroid, example is estrogen hormone, it directly crosses the plasma membrane, comes and binds to the heat shock proteins, okay, nuclear receptor which is bound to heat shock proteins, okay. Nuclear receptor, when it is in inactive state, it will bound to heat shock proteins. It comes and binds to the nuclear receptor. Immediately, the heat shock proteins get separated, you can see here, and there will be nuclear receptor This nuclear receptor will form a dimer, okay, NR dimer, and through this dimer, through the nuclear pore, enters the nucleolus, enters the nucleus and directly binds to the DNA. And there will be change in the replication of DNA, finally leading to change in the cell function. Understood? Uh, Ligand, hormone or estrogen crossing the plasma membrane binds to the type 1 nuclear receptor, which is bound to heat shock proteins, and when it binds, immediately heat shock proteins get separated, and the nuclear receptor, it forms a dimer. It passes through the nuclear pore and gets attached to the DNA, changing the transcription and translation process. In type 2, the where are the nuclear receptors present? Inside the nucleus, directly inside the nucleus. Okay. So the hormone crosses the plasma membrane, crosses the nuclear pore, directly goes to the DNA and binds to the receptor. Okay. When this receptor is in inactive state, it is bound to copressor. Okay. Copressor means it is the one which is going to suppress the receptor. But when the ligand comes and binds to the receptor, this copressor will be replaced by the co-activator. And finally, there will be change in the sequence of events. Okay. The receptor, the hormone crosses the plasma membrane, crosses the nuclear membrane, crosses the nuclear pore, enters the nucleus, goes to the DNA, binds to the receptor, and this receptor, when it is in inactive state, it is bound to copressor. It is bound to copressor, and when it is bound to copressor, once it is activated, the copressor gets replaced by the co-activator, and there will be change in the sequence of events. Okay, so this is about the cell, its structure, uh, different organelles in the cell, their function, then transport across the cell membrane, and finally, cell junctions, and finally, cell signaling pathways. So if you have any doubts, you can ask me. Yes, so thank you for attending the session. If you have any doubts, you can comment in the section. I will reply for it. And I request all the students to join or to subscribe the Prakara learning program. You have multiple options of notes, multiple options of video lectures. They provide you the best knowledge. So I request everyone to subscribe to Prakara Learning. And thank you for giving me this opportunity. Thank you for your patience. Thanks for listening. And okay, bye. Thank you all.